Scientists at ETH Zurich have pushed the boundaries of protein interaction research, enhancing a technique to study how proteins within cells communicate and collaborate. By improving a method called lip mass spectrometry, they now have the ability to analyze thousands of protein interactions, offering deeper insights into cellular function and disease. This advancement is critical, as many diseases, including cancer and Alzheimer's, are rooted in faulty protein interactions. Imagine a bustling dance club inside a cell, hundreds of proteins moving, interacting, and connecting with one another. Some connections are fleeting, while others last a lifetime, forming complex molecular machines. Under normal circumstances, everything flows smoothly, but when stress hits, proteins may switch partners, throwing the entire system off balance. Take, for example, protein red and protein blue. In healthy cells, they interact harmoniously, but under stress, protein blue starts to associate with protein yellow, a troublemaker that disrupts the cellular party. This shift in interaction is the root cause of many diseases. However, with the right medication, this unwanted partnership can be blocked or reversed, restoring balance. Researchers have also discovered that one key protein complex, SAGA, acts as the DJ in this molecular dance. When they removed SAGA, 2/3 of the other proteins stopped interacting normally under stress, highlighting how one player can influence the entire system. This discovery opens the door for pharmaceutical researchers to develop drugs that target specific protein interactions, potentially offering new treatments with fewer side effects. The journey of protein interaction research is far from over. The next step is to adapt these techniques to human cells, mapping their entire interaction network, or interactome. By doing so, scientists hope to pinpoint the exact moment when interactions go wrong, paving the way for innovative diagnostics and treatments for complex diseases like Alzheimer's and cancer. These advancements are already catching the eye of pharmaceutical companies. By targeting specific protein interaction sites, researchers can develop compounds that interrupt unwanted connections or strengthen beneficial ones. This represents a promising new frontier in drug development, offering hope for treatments that not only restore cellular balance but do so with fewer side effects than current therapies. Thanks for watching. If you are new to our channel, please subscribe to our channel and turn on the bell for further notification.